We now move on to parametric mapping, which is the application of kinetic modeling in every image voxel of a dynamic PET series. This creates new 3D images in which the voxel values directly represent a quantitative parameter from the model. For example, instead of kilobecquerel per cc or SUV per voxel in an FDG PET, the result of parametric mapping could be an image with quantified glucose metabolism in micromole of glucose per minute per 100 gram of tissue per voxel. Beyond interesting image presentation, parametric mapping allows image-wise averaging, subtraction. This can be within study groups or it can be for longitudinal measurements for single animals. An interesting example is shown here, whereby applying parametric mapping by calculating the transport K1 of the tracer compared to binding to receptors, 5-HT1A receptors in the hippocampus. The authors of this study in neuroimage were able to show the difference between two doses of teriquidar opening the blood-brain barrier. So we see an increase in tracer transport and a slight increase in receptor binding. In this exercise, we'll calculate parametric maps of glucose metabolism, and we'll use PATLAC graphical analysis. This efficient method linearizes the time activity curve in each image voxel, combining the time activity curve from the tissue with the blood data, the plasma input function, linearizing the data, and then a linear model can be fit to the portion of the data that is sufficiently linear. Because this is a simple linear fit, it can be efficiently applied across a whole image for parametric mapping. We do need blood data, and we do need the plasma glucose concentration in order to fit the slope in each voxel and convert it to glucose metabolism. Parametric mapping takes place in the pxmod tool. This tool is also organized with main tabs, where we set the model that we will be working with, and then within that tab we have sub-tabs taking us through the workflow. On the main tab itself, we can select the model that we will work with. In this case, we select the MR-GLUE FDG PATLAC model. Once this is selected, the sub-tabs and the settings that will be available within them are configured accordingly. For additional information about the model, you can use the question mark to reach our online documentation, and more information about that is given in section 9. Now we define the image data that we will work with using the Input Data Settings button on the right-hand side. In this dialog, on the top line, we select our data. In this case, we will use the database, and we'll use the Browse button on the right-hand side to open the database browser, where we'll select the Bruker PCI database. We can search for the rat subjects by typing in rat. And then for rat3, the dynamic FDG brain example, we can select the dynamic PET whole body brain crop series and set that series. We don't need to perform any initial pre-processing. We can use reset loading parameters to make sure that none is applied, and then with OK we can open the data. The load button itself is used when we're running through a protocol again, and we don't need to click it again now. Now we see our dynamic PET data. We can scroll to the last frame and adjust the upper threshold of the color table to see the same data that we saw in an earlier exercise. Next, we use the red masking button in the lower right to proceed. Now on the masking tab, we'll define a mask of the brain in order to limit the modeling calculations to the voxels that we're interested in other words, within the brain. Creating an average image that resembles the whole brain can therefore aid this threshold-based masking, and such an image can be created from a time range where tracer concentration is high throughout the entire brain. For FDG, this is late in the study when the tracer has been trapped in the brain tissue. Therefore, we can set the frame range to 20 to 23 and create the average image using the AVER button. The average image is then previewed on the average tab in the top left, where we can also adjust the color table and the thresholds to see our general brain outline. And we can use the data inspector to explore that image and find out what threshold or range of thresholds we might use for masking. The data inspector reveals that within the brain we have values in the range 150 to 200, whereas if we want to avoid the activity from the Harderian glands, we might choose an upper range threshold of around 300. Therefore, we can change the thresholding method in the lower left from threshold to in range. We can set the lower part of the range to 100 kilobecquerel per cc and the upper to 300 kilobecquerel per cc. Then preview the segmentation result using the segmentation button. The resulting mask, seen on the top right, has a rough outline, but it successfully excludes many voxels outside the brain. The mask should be saved to the database for later use in the protocol. We can edit the info before it's saved to the Bruker PCI database to indicate whether this was from our test run or whether we want to keep the original data. Once the mask has been saved, the warning message disappears and we can proceed to the next step. In this case, to apply the PATLAC model across the brain, we don't need to define any specific time activity curves, so we can proceed using the modeling workflow button. This takes us directly to the modeling tab, where we have the blood pre-processing tab. All we need to fit the model in all the voxels is our blood data and a fixed T-star value, which we will see in the next stage. First, we should provide the blood data, and this should be the arterial plasma tracer concentration throughout the dynamic PET measurement. For this data, we had the whole blood concentration and a fraction to convert that concentration to plasma values. And when that was done in the kinetic modeling tool, the resulting plasma concentration curve was saved for parametric mapping. Now it can be set in the input curve section, selecting time activity curve in the database, and we can use the browse button to find for RAT3 the calculated activity in plasma. Now that's set to our protocol, meaning that we can proceed again with pre-processed blood. On the blood results tab, we see a preview of our blood data, which we should recognize from the kinetic modeling stage. Next, we can continue straight away with the configure model processing button. Here we have some more settings for the particular model we're working with. You see that target tissue is not active because we skipped the time activity curves and VOI's process. That does mean that we need to fix a T-star rather than calculating it from a preview time activity curve of the data. The T-star can be fixed by unchecking the checkbox, and we can enter a value of 15 minutes based on our prior knowledge from kinetic modeling. On this page, we would also enter the individual plasma glucose, which you should measure for each rat when you're working with FDG in this way. Now we can proceed using pre-process model. We see no particular model results because we didn't provide a time activity curve, but that's okay. We speed up the process. Then we move on by configuring the target maps. In this case, our map for the metabolic rate of glucose consumption. The pixel-wise calculation can then be started using the workflow button in the lower right, Start Pixel-wise Calculation. The resulting parametric map of glucose metabolism can be explored using the data inspector. Note now the quantitative units, micromole per minute per 100 gram. The maps can then be saved in one of the available formats, for example back to the database, so that we have our study organization in the easiest way. Again, we select the database we want, and we can see that in the series description, PMOD automatically indicates what kind of parametric map is being saved. We can also use the save icon on the right lateral taskbar, depending on the process you want to use. We highly recommend saving the full processing configuration in order to retrieve all of the processing details later. This is achieved with save protocol in the status line. And we can save this run through the tool. Indeed, it is FDG PET with no target region defined, and our test run for the exercise. To test the configuration, we can open a new Pixmod workspace, the Add Tab button at the top of the tool. By default, this uses the same model in the previous tab. Next, we can use Load Protocol at the bottom of the tool and select the protocol that we just saved. We see that it knows which data to work with and which mask. We can allow it to execute and then run the protocol. Each step is performed in sequence, and then we have our parametric maps rather quickly.